exercise 3 is all about a particular method that we use to count cells, and that's called the viable plate count method. So first, what I want to do is think a little bit about why we would want to count cells in the first place, give you some examples there. I want to talk about a couple of different ways to count cells that we would commonly do in the lab. And then I'm going to talk about the viable plate count in particular and explain how that procedure works. And we also have to go through how to do some calculations. So I've got this split into 2 different videos. So why would we want to count cells? Well, let me give you a few examples. In the clinical lab, a patient is only considered to have a urinary tract infection if they have above a certain number of cells. And the number that I found is about 100,000 cells or colony-forming units. We'll talk about what that means in a minute, per mil of urine. Okay, so in the clinical lab, it might be important to get a count of cells to see if a patient has a UTI. Lots of applications in public health, testing our food supply. Milk, for example, is commonly tested both right when it comes from the cow, we would call that raw milk, and also after it's pasteurized. So we have to keep those counts below a certain threshold in both cases for it to be considered, safe for most people to drink. And then in the research lab, it might be important for the researcher to know about how many cells they have in a sample. It's often important to know if that cell population is actively growing. You might be comparing growth rates in certain conditions, or some cells just act differently when they're actively growing. So, for instance, bacteria often only produce certain enzymes when they're actively growing. They're most susceptible to antibiotics when they're actively growing. So there's a lot of instances, and there's just a few examples why it might be important to count cells. Okay, there are a lot of different methods to count cells. I want to just compare our method in class to a couple of other ways. Our first method here is using a hemocytometer. And that's kind of hard to read that, but that's written right there. This is also called Petroff-Hauser chamber. Okay, I usually refer to it as a hemocytometer because that's just a little bit easier to remember. So the way this works, you actually have a suspension of cells that you add to this big glass slide. You add it right here, and then actually you have a cover slip over it like this. And so when you add your suspension of cells by, say, a Pasteur pipette or a micropipetter, it wicks underneath there, and then you have kind of a even layer of your cells. You'll be able to see that a little bit better right there. And again, there's going to be a cover slip right here, which is kind of either not there or hard to see. All right, so basically right here and right here on this slide, there's etched a grid, and you would only see that if you were looking through a microscope. And then you actually count a certain number of squares in that grid, how many cells are visible. So in this example, that would be fairly easy to do. You have a nice countable range of cells here, and you take the number that you count and you mathematically can extrapolate back to how many cells were in your original sample. Some drawbacks with this is that these are probably some kind of yeast cell or blood cell because they're pretty large. Bacterial cells are a lot smaller, a little bit harder to count, and definitely harder to tell just by looking at them if they're dead or alive. And oftentimes it's the ones that are alive that we're really interested in. Okay, and you also have to have, again, the right dilution If you've got too many here, you can't count them accurately. If you've got too few, it's not statistically significant. Okay, but that's direct cell counts using a hemocytometer. Another method, you hopefully are a little bit familiar with this instrument. It's a spectrophotometer. Hopefully you use that in Bio 120 and maybe some other labs as well. Remember how a spectrophotometer works. You put your sample here in a cuvette, and that goes in the machine right there. Remember, a sample with bacterial media that's got bacteria growing in it would be cloudy to a certain extent. And so we can measure how much the light is absorbed or how much the light is transmitted. Okay, usually we measure absorbance. We've got a particular wavelength that we usually use when we're testing for turbidity, we would say. And the more turbid it is, the more light is going to be absorbed and the higher the absorbance would be. All right, so here's an example, right? You've got a sample where there's lots of bacteria growing, and here's one that's pretty clear and none or maybe very few cells growing. All right, so using a spectrophotometer is a really great way to see if your cells are growing over time. Of course, it gives us an absorbance number. It doesn't give us an actual number of cells. So you would have to, if you wanted to get actual cell counts here, it would have to be, you would have to do kind of an assay over time, and you would have to maybe take out your absorbance sample and then actually count that sample maybe with a hemocytometer and make a standard curve. Whoops. Wrong button there. Sorry. Okay, so here's numbers of cells on the X and on the Y and absorbance on the X. And then next time you took an absorbance reading, you could, okay, that corresponds to whatever number. But again, absorbance by itself is not of course, an actual number. All right, so to kind of think about comparing those methods with this is our viable plate count method. So I got this figure from your textbook. It's in chapter 6 that you talk about all different counting methods and cell growth in lecture. Okay, so with the viable plate count, you take what we normally in lab, we have some stock culture. And that's what we're trying to figure out, the approximate cell density, original cell density. So original cell density of this stock culture. Okay, so typically if we were to just take out some of that stock culture and put it on some solid media and spread it around, you're going to have just, way too many colonies growing here. So too numerous to count. Okay, so we almost always, if you're taking, certainly if you're taking a sample from nature, if you're growing cells up in the lab, there's almost way too many there, and we have to dilute it out in order to get a reasonable number to count. Okay, so we take out an aliquot. In lab, typically we'd make 1 to 10 dilutions. I'm going to talk about how to make dilutions in a minute. But we make serial 1 to 10 dilutions. Okay, so typically the way that would work in the lab, the first person would make their dilution and then streak out an aliquot. We take 1/10 of a mil to put it on our spread plate, pass that on to the next person, and they make the next dilution. So we have rows of 5 work together And that way everybody only has to make one dilution rather than everybody doing all 5 and everybody having 5 plates. So typically in this lab we work together. 0.1 mil is generally about a drop or 2. Okay, so we take that out with a pipette and just measure it as accurately as we can. We do what's called a spread plate. We should have practiced that the very first lab, back in lab 2, where basically you have to use this glass hockey stick, we call it, to spread our cells around evenly on the probably tryptic soy agar plate, for example, or nutrient agar plate. And that's generally one of the things that students enjoy the most because, of course, you have to get this glass hockey stick. You have to sterilize that, so we do that by dipping it into some alcohol, catching it on fire, letting it cool down a little bit, and then using it to just kind of spread those cells around as best we can. Okay. Then we incubate. At this point, incubate overnight, and the next day you come in and count the number of colonies that grow up on the plate. And we need a reasonable number to count. We look for between 30 and 300 colonies in order to do our calculation. So here it looks like where the cells were the most dilute, we're going to have too few to count. Here it looks like 1, 2, 3, 4, 5, 6. That's also going to be too few to count. Okay, looks like maybe this plate here might be kind of a reasonable number. I have students in class maybe split that plate up, right with their Sharpie on it, and maybe count 1/4 of it, and then, multiply up to get the full number. Okay, and then again, in the next video, I'm going to talk about how to actually do those calculations, but it's a pretty simple procedure actually to do this in lab. All right, so just thinking big picture, once again here, comparing those 3 methods those 2 methods to the viable plate count. If we think about, well, what are the advantages of using the viable plate count method? Think about the materials that we use. They're pretty inexpensive. Okay, we don't need a microscope, we don't need a spectrophotometer, we just need, some media and some petri dishes, a warm place to incubate maybe, and really it's very cheap. The materials are easy to obtain. Okay, and another big example, the V in viable, only live cells are counted. Okay, it's only going to be a live cell in that original sample that actually grows up into a colony. Of course, that one cell, when they grow exponentially, it very, very quickly becomes 1 million, and of course there's about 1 million cells in a colony. Okay, if this cell in the original sample is dead, it's not going to grow into a colony. Okay, so only the live cells are counted, and that's usually a good thing. Okay, there are a few disadvantages. Of using the viable plate count to determine cell numbers. Hopefully, one of the big ones that comes to your mind right away is the time. Okay. Viable plate count requires an overnight incubation. Okay, it doesn't take long to actually, inoculate it and make the dilutions, but then you have to wait for the cells to actually grow up. And that might be fine, that might not be fine. Okay, Also, making dilutions are necessary. Now, in reality, that's often necessary for a hemocytometer and for using a spectrophotometer as well. Okay. Another disadvantage, cell clumping can lead to an underestimation or undercounting cells. Okay, so basically we'll talk more about this when we're talking about our calculations, but there are some kinds of cells, like for instance our staphylococcus, that tend to grow in clumps, and sometimes those clumps don't break up. You might have, a clump of cells here instead of just one cell, and you might have actually a colony grows up from a clump of cells. Okay, so that's going to lead to a little bit of an underestimation to the actual cell count in this original sample. Okay, so when we do our calculations, we use units that kind of remind us of that. Okay. Okay, so hopefully you understand a little bit about how you would inoculate the viable plate counts, and in terms of what's the best way to count cells, it really depends a lot on what kind of equipment you have and how accurate your counts need to be, how much time you have, lots of different things, okay? So next video, we're going to talk about a little bit of dilution theory, important life skill to have, as well as how to do these calculations.